If the post-war years have been characterized by the name Green Revolution, the end of the 20th century might be considered as the Blue Revolution era. Indeed, the past 20 years have seen the accelerated development of a new discipline in the farm produce industry. This discipline is aquaculture, that is, the production of aquatic organisms for consumption, preservation and repopulation ends, as well as for sport fishing purposes. Nowadays, over 2,000 species of fish are being farmed, either on an experimental or a commercial level, to which should be added algae, mollusks and crustaceans. Pisciculture, that is the branch of aquaculture concerned with fish, deals with both marine and freshwater species. Though rearing techniques may vary from one species to another, they aim in every case to control the various production phases. The purpose of this film is to give you a glimpse of the rearing techniques used in aquaculture. To illustrate this presentation, we've chosen the river perch, a freshwater fish currently being researched. This fine fish, with its greenish skin streaked with black and its brightly colored fins, is well known to sports fishermen and a few gourmets who particularly appreciate the quality of its flesh. The perch can indeed be considered as one of the best freshwater fish of these regions. Domesticating this species involves a full control of its various rearing phases. Moreover, these different phases will constitute the main theme of this film, namely breeding, larvae rearing and fry production, and finally fattening until the commercial stage. In aquaculture, breeding can be defined as the production of viable larvae. It constitutes a key phase in the intensive production of a species, and very often determines the development of the later stages of rearing. However, it may happen that the breeding process is not properly mastered, and it therefore becomes necessary to resort to taking fry from their natural environment. In this type of fish farming, the sires are, as is generally the case, taken from stocks of fish that have been held in captivity for several generations, and from which an appropriate selection has been carried out. First, let's see how a perch's annual breeding cycle unfolds in a natural environment. In summer, the female is sexually at rest. She needs to undergo a period of low temperature during which her abdomen swells through the accumulation and development of the oocytes. She's then ready to lay in spring when it starts to get warmer. This information is of prime importance if one wishes to someday obtain sires capable of breeding in captivity at any time of the year. The keeping of selected sires in a closed circuit with controlled modification of temperature and light cycles is an increasingly used technique in the commercial fish farming. It's currently being experimented with on the perch. Thus, an artificial modification of the temperature cycles should allow the mating season to be brought forward or delayed, and for a female's annual number of layings to be multiplied. This modification of the temperature cycle is usually combined with a modification of the photoperiod, that is, the night length-day length ratio. Once mature, the perch, whether wild or domesticated, lays very easily and spontaneously in captivity. This constitutes an undeniable advantage in fish farming. Once the temperature reaches at least 12 to 13 degrees Celsius, the female lays a long string of eggs that are linked together by a gelatinous substance that gives the whole a stretched accordion-like look. As we will see at the top of the picture, as soon as the eggs are laid, they are rapidly fertilized by the males. Spontaneous reproduction on the part of the sires often occurs randomly. Under certain circumstances, such as the need for fertilized eggs at a specific moment, the synchronization of several females' layings, or the production of polyploid individuals, it's necessary to resort to a controlled triggering of the laying, followed by an artificial fertilization of the eggs. In order to achieve this, the males and females undergo hormonal treatment in the form of an intraperitoneal injection of synthetic neurohormones or hypophysial extracts. The fish are then separated according to sex and kept undisturbed to avoid spontaneous breeding. A few hours, usually a full night, are necessary for the hormonal treatment to induce ovulation in the female and an increase in the volume of semen in the male. After this latency phase, the male will emit a few milliliters of sperm by having its sides massaged. With the female, a massage of the abdomen will cause the ovules to be expelled. The sperm is then immediately distributed over the ovules. The eggs are then carefully arranged in incubators where they will stay shielded from light until hatching time. Incubation constitutes the period during which the embryo progressively develops in the egg by feeding on the reserves of yolk accumulated by the mother during the months preceding the laying. This embryo is easily visible through a magnifying glass a few days after fertilization. The length of the incubation period depends on the temperature of the water. Thus, this phase lasts up to a month at 6 degrees, 12 days at 12 degrees, and only 4 days at 20 degrees. This variation in the incubation period's length according to temperature can be used to accelerate or, on the contrary, delay hatching. On hatching, the perch larva has a small vitelline vesicle that enables it to survive for a few days without a supply of exogenous food. This vesicle will rapidly be reabsorbed within 3 to 4 days, and the larvae will then be forced to find food in the external environment. With the perch, larval rearing constitutes the most delicate phase, for three reasons. First, because this phase is characterized by the production of small-sized eggs, where the larvae will not exceed 5 or 6 millimeters in length on hatching and will weigh less than a milligram. Secondly, their dependence on a natural diet made up of living prey, of which the size must be inferior to their oral cavity, also constitutes a limiting factor for successful rearing. And finally, added to these constraints, is the perch's cannibalistic behavior during the first few months. Larval and fry production are the object of research that aims at comparing the efficiency of farming techniques. The tested techniques differ according to their degree of intensification. That's to say, the quantity of fish produced per surface or volume unit. There are three techniques: the extensive system in fertilized pools, semi-intensive fry rearing in fertilized ponds, and modern intensive larviculture techniques in raised tanks. The quantity of fish increases as we pass from one technique to the other. We shall be looking at each of these techniques. A few days, usually a week, before the larvae are tipped into the pond, the latter is inorganically or organically fertilized. This fertilization ensures an enrichment of the water, mainly in nitrate and phosphor, nutriments that are essential to the development of an abundant phytoplanktonic biomass. Thanks to this abundant phytoplankton, which is responsible for the water's green coloring, the zooplanktonic organisms, such as rotifers to start with, will, if sunlight and temperature conditions are favorable, be able to develop and reach high densities by the time the fish larvae hatch. These phyto and zooplanktonic densities are checked before the eggs are tipped into the pond. One day before the larvae hatch, the eggs are arranged inside small floating cages fitted with meshes that hold the eggs in but allow the passage of larvae after hatching. The larvae will rapidly colonize the pond by feeding on rotifers and subsequently on the Cladocera and Copepoda present in the environment. A regular check of the phyto and zooplanktonic densities can be carried out and a new fertilization performed if necessary. Regular sampling of larvae and fry enables their growth to be assessed. This sampling, performed with a square fishing net, determines the quantity of food the fish should receive. Three to 6 months, according to the desired size after immersion of the eggs, the fry are collected by emptying the ponds completely. In the case of the perch, the semi-intensive system is made up of relatively large tanks, from 10 to several tens of square meters, in which several thousands of eggs per square meter are immersed one day before hatching. A week before immersion, organic fertilizing, similar to that performed on the fry ponds, will have favored the development of a large rotifer population. These rotifers serve as food for the perch larvae during the days following their hatching. Because of the high larval density, the stock of rotifers will rapidly be consumed. That's why the larvae are fed Artemia nauplii, a small planktonic crustacean, several times a day, a few days after hatching. These Artemia nauplii constitute an excellent substitute food for the zooplanktonic organisms naturally present in the diet of young perches. At the end of the first week, artificial food is given to the larvae as a complement to the Artemia nauplii. This artificial food is intended to progressively replace the Artemias during the weaning phase. It must meet the larvae's nutritional needs as closely as possible. About 40 days after the eggs are immersed in the fertilized tanks, the fry are collected with a square net at an average size of 0.2 to 0.5 of a gram. After being collected, the fish are carefully sorted so as to separate the very large individuals, generally cannibalistic, from the normal size population. Cannibalism can indeed constitute a major cause of mortality. With intensive farming, the initial density of larvae can reach very high figures, varying between 50,000 and 200,000 larvae per cubic meter, according to species and development stage. The quality of the water and the hygiene of the farming environment are very important parameters to be mastered. That's why intensive larval farming is often carried out in a closed circuit, with recirculation and physical and biological purification of the farming water. Another primordial factor is the quality of the food fed to the larvae at their earliest age. 2 days after the larvae's massive hatching, that is before their vitelline vesicle is completely reabsorbed, the larvae are fed with Artemia nauplii. At this stage, the oral cavity is inferior to 400 microns, which prevents a part of the perch population from ingesting the Artemia nauplii. A complementary supply of rotifers, of which the size barely exceeds 100 microns, enables the smaller larvae to feed for a few days until their oral cavity becomes compatible with the size of the prey. As the larvae have a very short digestive tube, the intestinal transit time will also be very short. In order to increase the efficiency of digestion and assimilation of food within the larvae, the fish will be fed very often in the course of the day, preferably with an automatic feeding system, such as a peristaltic pump, that continuously delivers small quantities of live food. At the end of the fry stage, and after a weaning similar to the one described in semi-intensive farming, that is, by progressive reduction of live food supplies in favor of artificial food, the young perches are transferred, after weighing, to the fattening tanks. The fattening stage constitutes the last production phase of a species. This period begins as soon as the fish is past the fry stage and ends with the obtaining of commercial size fish. With the perch, this size varies according to the regions where it's commercialized and consumed. From 120 to 150 grams in alpine countries, up to older fish of 300 to 500 grams in the north of France, Belgium and Germany. We shall now look at the final operations to be carried out in order to obtain the desired result. During the fattening phase, food generally constitutes the major part of production costs. It's therefore vital to give the fish a food that meets as closely as possible the species' nutritional needs. From a qualitative point of view, the diet must be balanced. This balance takes into account a few important elements such as the protein-energy ratio level, the rate of lipid contribution, the composition in amino acids and essential fat acids. From a quantitative point of view, the objective is to cover all the fish's maintenance needs and to allow it to attain optimal growth. The alimentary ration depends on numerous factors, such as the size and age of the animal, but also on rearing temperature. The quantity of food to be distributed is lower with large fish farmed at low temperatures. To illustrate this, here's some information concerning the fattening of the perch. The fish's growth and the production of a farming tank are also influenced by environmental and social factors, such as stockage density, heterogeneity of individual size, and the relation between dominators and dominated in the group. In order to reduce this difference in size between fish from the same tank, and thus the hierarchy that results, one must regularly sort them. This allows a more homogeneous growth of the whole stock. Establishing all these biological and zootechnical parameters results in better growth of farmed fish and a better use and transformation of the distributed food. As we've just seen, the domestication of a species in aquaculture requires the full control of several stages. First of all, the sexual maturation and egg-laying of adults in captivity during the normal breeding period or otherwise. Next, the farming of larvae in more or less intensive and complex conditions. And finally, the fattening of the young fish until they reach commercial size. Each production system has its advantages and limitations. Apart from the understanding of each of these stages, the rigor and care with which the techniques are applied helps to improve the zootechnical performances of the species. With a newly domesticated fish, such as the perch, this improvement can only be acquired through the progressive selection of clones and individuals over the passing generations.